Hi, I'm Patrick Shih. I'm a grad student in Cheryl Kerfeld's lab at UC Berkeley. And I'd like to thank the organizers for giving me a chance to talk today about a collaboration that's been ongoing between the Pasteur Culture Collection in Paris and the Joint Genome Institute's GEBA initiative, which stands for the Genomic Encyclopedia of Bacteria and Archaea. And the GEBA project is aimed at improving the phylogenetic coverage of bacteria and archaea by sequencing genomes in sparsely covered regions of the tree of life. So in the same spirit, our CyanoGEBA project is aimed at improving the sampling of our favorite phylum, Cyanobacteria, which are here on the tree of life. So why Cyanobacteria? Cyanobacteria are incredibly diverse. This can be shown through the multitude of morphologies that can be found in this phylum. They can also be found free living in aquatic to terrestrial environments, as well as symbionts of marine animals and of plants. Furthermore, they've contributed greatly to eukaryotic diversity through plastid endosymbiosis, and they're an incredibly DOE relevant bacterial phylum because of the many different metabolic pathways that they have, seen here from their these protein complexes. So they have the ability to fix nitrogen, if we can figure this out, produce hydrogen, and most importantly, they are the inventors of oxygenic photosynthesis, which is this photosystem right here. And as the inventors of oxygenic photosynthesis, they gave rise to the Great Oxidation Event 2.5 billion years ago, which is responsible for all the oxygen we have in the atmosphere today. So there's a lot of reasons for why we should sequence more cyanobacteria, but today I'll be talking about just 3 stories that we've been looking at. First, the implications on morphological transitions. Secondly, our understanding of endosymbiosis. And thirdly, how this affects our knowledge of photosynthesis. So the Cyanobacterial taxonomy has classically been based on morphology. There are 5 morphological sections, section 1 being unicellular, and they divide through binary fission. Section 2 are unicellular and can divide through multiple fissions. Section 3 are filamentous. Section 4 are filamentous and have the ability to undergo cell differentiation to make specialized cells called heterocysts, which allow for nitrogen fixation. And section 5, which are filamentous, they have heterocysts, and the filaments actually have the ability to branch. So prior to this study, there were no sequenced genomes from the section 2 or section 5. Like I said, there are no section 2 or section 5 genome sequence. However, from the CyanoGEBA study, we've actually sequenced 54 cyano-genomes, 6 of which from the section 2, 5 from the section 5, and we're actually sequencing genomes across all the morphological sections. So as a visual representation of how we're improving our phylum coverage, here are all known 16S cyano sequences from the Green Genes database prior to the study. Set at a 95% identity threshold, we get a little more than 100 clusters. And what's important here is we've highlighted in blue all the clusters with at least 1 sequence representative. And more importantly, the branch width represents how many sequenced genomes are in that cluster. So you can see here in this marine Prochlorococcus sinococcus group that you heard about earlier today, they're incredibly globally relevant, but almost half of the sequenced cyano-genomes are from this one cluster. So this is really biasing and skewing our understanding of the entire phylum. So once we add our CyanoGEBA genomes, you can see that we're really improving the phylum coverage. And ways that we can quantify this is by using certain metrics such as phylogenetic diversity and tree imbalance. So the phylogenetic diversity metric measures the length of branches in a phylogenetic tree. So if we compare the CyanoGEBA taxa versus the publicly available genomes, we can see that we're getting roughly a 2-fold improvement in our coverage. Other metrics such as tree imbalance measure how equally distributed branches are on the tree and can serve as a proxy of improved coverage. So when we compare, again, the publicly available genomes compared to when we add our genomes, we can see a decrease in imbalance, which means that we're getting better coverage of the entire phylum. So now that we have full genomes, one of the first things we wanted to do was to make a robust species tree. So we took 31 conserved proteins and we concatenated them, and we got a nice species tree, and we mapped on the morphologies onto it. And what this showed was that there was a convergence of morphologies. So if you just look in the light blue, you can see that this filamentous phenotype has arisen independently several times across the entire cyano tree. Likewise, the section 2, or these unicellular cyanos that can divide through multiple fissions, also have arisen independently at least 3 different times. So that shows that these morphological phenotypes, the filamentous and the ability to divide through multiple fissions, are relatively easy to be gained or lost. However, other morphologies, such as the ability to undergo cell differentiation to make these heterocyst cells, are much more complicated. And that's displayed here because that characteristic has only been gained once in the entire cyano tree. Furthermore, the ability to have branching filaments also seems to be relatively complicated, as this has only arisen once. So because we have full genomes, we can actually begin to do some comparative genomics to try to understand what are the underlying genetic elements that are responsible for allowing for these morphological transitions. So here I'd like to point your attention to the top, where there's 3 filamentous cyanobacteria and a reversion to unicellularity. And when we look at the genes that are different between the filamentous and the unicellular cyano, we can actually find that there's 121 genes, 90 of which have predicted transmembrane helices. Now, this is really interesting because normally we're taught that roughly 10 to 20% of all genomes are membrane proteins. However, we see a very clear enrichment of membrane proteins in these 121 genes. And this is very analogous to what's been studied in the eukaryotic world studying multicellularity, because it's been known that protein families such as cadherins and adhesions are really important for allowing these cell-cell interactions, allowing the cells to stick to one another, allowing for multicellularity. So likewise, when we look at another test case, another reversion to unicellularity, we see, again, another enrichment in membrane proteins. So the next major thing we were interested in is plastid endosymbiosis. And this is because it's driven a lot of eukaryotic diversity, and it's considered a major transition in life. So in a nutshell, it's thought that a eukaryote ate a cyanobacteria, which gave rise to all plastids, which is the Archaeplastida lineage. This event is called primary plastid endosymbiosis. There were actually subsequent secondary endosymbiosis events of green algae to give rise to the euglenoids and the chlorarachnophytes, and another secondary endosymbiosis of a red algae, which gave rise to the entire chromalveolate lineage. So we wanted to ask the question, where do all plastids fit within the cyano tree? And in order to do that, we took 25 conserved plastid proteins, concatenated them, and compared them to our improved cyano data set. So here's that tree, and you can see that all plastids are monophyletic. And here we have our primary plastid endosymbiosis and our basal glaucophyte lineage. We have our red algae and their secondary endosymbiosis to give rise to the chromalveolates. We have the green lineage and their secondary endosymbioses. And the punchline is, it seems that primary plastid endosymbiosis happened relatively early in the history of extant cyanobacteria. This is displayed by the very deeply rooted branch here, that plastid branch, and how it's very deeply placed in the entire cyano tree. So the next thing that we were interested in is, now that we have all these new cyanoproteins, can we improve our understanding of endosymbiotic gene transfer? Endosymbiotic gene transfer is when genes from the original endosymbiont, or cyanobacteria, were transferred into the nuclear host genome. And because we have a more complete data set of cyanoproteins, we can have more accurate predictions of what eukaryotic proteins were actually of cyano descent. So when we look at eukaryotic nuclear genomes that have already, prior to this, have been sequenced and compare them to all cyanoproteins before and after we add our CyanoGEBA data set, we clearly see that there's always an increase in the amount of proteins that are actually now predicted to be of cyano origin. So finally, I wanted to talk about how this data set affects our understanding of photosynthesis. And specifically, I'm going to talk about cyanobacterial light harvesting complex diversity. So traditionally, it's thought that cyanobacteria have soluble pigment protein complexes called phycobilisomes. These are these large rod-shaped soluble complexes that absorb light and funnel them to the photosystem. However, plants have membrane-bound light harvesting complexes. However, in a handful of cases, we know some cyanobacteria actually have membrane-bound light harvesting complexes that are called CBPs, or chlorophyll binding proteins. So here's a cryo-EM of the photosystem, and when these CBP proteins are induced, you actually see them associated along the periphery of the photosystem. And this is thought to be a unique strategy for some cyanobacteria to help absorb light, other than using phycobilisomes. So with a more comprehensive cyano data set, we actually find that these membrane-bound CBPs are not a rarity in cyanobacteria, but rather many cyanobacteria have these CBP proteins. More interestingly, given the larger data set from the CyanoGEBA genomes, we actually find new, novel, uncharacterized clades of these CBPs, which could potentially be allowing cyanos to have diverse and unique methods other than phycobilisomes to absorb light. So in conclusion, cyanobacteria may emerge as a model phylum for understanding morphological transitions. They can improve our understanding of endosymbiosis and photosynthesis, and CyanoGEBA genomes provide an unprecedented amount of information about cyanobacteria metabolism with implications for bioenergy research. So I'd like to acknowledge our collaborators at the PCC, Muriel Gugere, Rosemary Ripka, Nicole Tandudu-Mersak, our collaborators at Davis, Dongying Wu and Jonathan Eisen, my advisor Cheryl Kerfeld and the entire Kerfeld group, and all the people at JGI and Los Alamos that helped with this huge genome sequencing project, and my co-advisor Chris Niogi, and NSF and DOE for funding. Thanks.
Thank you, Patrick. One question, again, before we break. I have a quick question. How important was the availability of complete genomes for this project versus having draft for the work you have been doing?
Complete genome?
Yeah, the availability of complete genomes. Most of the genomes you have been...
So it just depends on the question that you're asking. If you're trying to compare genomes between each other, it's difficult to have a draft genome when you want to, say, look at all heterocyst cyanobacteria, but say a quarter of them are of draft or in a draft shape. So you can't really say what are all the genes that are necessary for heterocyst cyanos now. But for specific phylogenetic questions, I think the draft genomes were perfectly fine.
But for many of the questions you had, the availability of the complete genome was a key.
Yes, definitely.
Okay, thank you. I would like. Oh. I can't say no.
How many do you think would be enough in terms of additional sequences?
So if you look at just based on 16S, we are getting much better coverage. However, there's still a lot of clusters that have nothing near it, and this area in general. So arguably, it's not like we need to throw. There's not like a finite number, like we need 200 more, but it's just we need to be more. We need to be smarter when we're choosing which genomes to sequence if we're going to tackle these questions of having a better coverage of the entire phylum. So if we had, I don't know, 5 of them from this cluster, I think that'd be more important than, arguably more important than sequencing more Prochlorococcus. Yeah.
Thank you, Patrick.